Thank you. Thank you so much.
Okay, we can start the topic. Okay.
Yes, thank you.
So we are going to see the pharmacological evaluation of medicinal plants and herbal products for another 1 hour. Okay. Um, in our modern medicine system, also the traditional medicine system, nowadays the people are depending natural products, plant-based medicine for to cure many kind of human ailments, especially the modern medicine system and therapeutic medicine system. They are depending the plant-based medicines for cure various kinds of diseases like cardiovascular disease, cancer, diabetic, and inflammation, pain, these kind of diseases. So the pharmacological evaluation of medicinal plants and herbal products are very important in modern and therapeutic medicine system in nowadays. Okay. Um, all the natural products and natural product based medicine therapeutic agents are developed from the biodiversity. What is mean by biodiversity? Biodiversity, nothing but it is a biologically variety and variability among living organisms and the ecological complexes in which they occur. So, biodiversity is the main part of the natural product. All the living organisms and non-living organisms and all the materials are available in biodiversity. So these are all living organisms is called biodiversity. It measures at three levels. Uh, First level is gene level and the species level and ecosystem level. These are the three levels are main part of the biodiversity. First one is genetic diversity. Genetic diversity means diversity of genes within a species. That means one single species have many different kind of variation, genetically varied, like a color, like a character, like a um, uh, character-wise changes, color-wise changes, and height. These kind of genetical variations in a single population. That means genetic diversity. And specific species diversity. Species diversity means biodiversity hotspot. In this diversity, species diversity have a lot of animals and birds and bacteria, fungi, virus, all kind of living things. This is the species diversity. And ecosystem diversity. Ecosystem ecosystem diversity means all these living things are present in different kind of ecosystems like terrestrial ecosystem, soil ecosystem, and water ecosystem, and mountain ecosystem. These are the different kind of ecosystem. All living things are available in these kind of ecosystem level. So the biodiversity level is three kind level. One is gene level, and another one is species level, and another one is ecosystem level. And
Sir,
it
I'm
is the
sorry to interrupt you.
yeah, yes, ma'am.
The slide is not passing for us.
Okay, I will show you. This is
Now pass it.
okay. This is ecosystem diversity, and this one is species diversity and genetic diversity. These are the three kind of steps are available in biodiversity level, measured in three levels. One is genetic diversity and species diversity and ecosystem diversity. Okay, and biodiversity is the natural asset. How it is possible? The biodiversity have many kind of products like food, medicine, materials, chemical products, water and soil supply, and climate regulations. And also the biodiversity which helps to the human beings life, recreations, to enjoy the sea, enjoy the beach, enjoy the nature. like recreations and inspiration. Biodiversity inspiration for others, and it gives the spiritual stimulation. It increases the stimulation of spiritual and also peace of mind. So biodiversity is a natural asset for all kind of recreation, inspiration, spiritual stimulation, peace of mind. It contributes to the social economic development of the society. So biodiversity is important for our human life, which brings many kind of useful, and it is necessary to uh, develop the status of biodiversity, and we need to hold the biodiversity level in our earth system, which helps for the future humanity. So we need to save we need to save our biodiversity to improve the future humanity. Uh, Already I told you this biodiversity, one of the important point to develop the natural products. What is natural product? Natural products are naturally available in living, naturally available in the biodiversity. The products which produced from the natural things is called natural products, like living organisms, especially plant-based natural products, animal-based natural products. These are the living things products. So the products produced from the living things also in natural is called natural products. So in my topic, I'm going to explain how the natural products used in our human medicine system, particularly plant-based medicine system. So the medicinal plants are having lot of natural products which improves our human ailment, particularly which control the diseases of the human beings, like many kind of ailments like cardiovascular disease and diabetic disease and hepatotoxic disease and um, anti-inflammation and also pain. All these kind of diseases controlled by the natural products. So the natural products improve the healthy life of our human being. And the medicinal plants having many kind of natural compounds. Especially the traditional medicine system, medicinal and aromatic plants are known to be used 70 to 80% of the global population for their medicinal therapeutic effect. These plant products serve as a sources of many drugs and therapeutic agents. Particularly, these plant materials having phenolics and flavonoids, terpenoids, these kind of compounds which have many kind of therapeutic effects. People are becoming highly interested in the medicinal plants because of the therapeutic performance and low toxicity. All these kind of medicinal plant-based drugs having low toxicity and higher efficacy for the disease control. Because these plant-based products having the secondary metabolites which generally produced by the plants for their defense mechanism. In this defense mechanism, plants uh, protect them with their diseases comes from the insects and other pollution controls. So this defense mechanism developed from the secondary metabolites of the plants. So these secondary metabolites which helps defense mechanism of the plants, including which gives the therapeutic agents like phenolics, flavonoids, terpenoids compound. These kind of compounds helps to control our human being different kind of diseases. It have been implicated in therapeutic properties of most of the medicinal plants. Secondary metabolites such as alkalides, phenolics, flavonoids, terpenoids, and saponins and iridide compounds. These are the main products of the secondary metabolites. Screening of plants for their active compounds leads to the innovation of new drugs. So, it is necessary to screen the plant-based active compounds, which is the very innovation for development of new drugs, which gives more efficacy to protecting the human beings with the harmful disease, including healing of wounds, inflammation, pain, diabetic, cardiovascular disease, gastrointestinal disorders, hepatic liver disease, cancer, and etc. Mainly, mainly these secondary metabolites, like phenolics, flavonoids, terpenoids, saponins, all these kind of secondary metabolites nowadays available in the commercial market. Commercial market. So it is already commercially market. So it helps to develop the new drugs in the modern medicine system. I'm showing here monoterpene, one of the terpene compound, which is mostly available in the menthol plant. So it helps to reduce the pain, inflammation, and also stress condition, relieve the stress condition of human beings. And sesquiterpene, artemisinin, artemisinin, it is one of the terpene compound, the same diterpene. All these compounds are nowadays available in pharmaceutical market, like a pharmaceutical drug companies, industries. All these industries produce this kind of medicine synthetically and developed in the marketization. And also flavonoid compounds. Some of the flavonoid compounds already in the pharmaceutical market, like silymarin. Silymarin, it is a compound which is used for cure the hepatic disorders, especially hepatoprotective activity. This compound having hepatoprotective activity, so it cure the liver disease. And resveratrol. Resveratrol is the one of the compound which is also used to cure the cancer kind of diseases. So all these flavonoids are available in the pharmaceutical market nowadays. What are the fats? What are the fats which is used in the plant-based products? Plants have provided a source of inspiration for novel drug compounds. I told you already, novel drug compounds means phenolics, flavonoids, terpenoids, saponins like compounds. These are the novel drug compounds available, plenty of compounds available in the plant system. Medicinal plant research currently receiving increasing interest from the academia and industry. Why? What is the necessary? What is the necessary medicinal plants research currently increasing interest? Why means these medicinal plants having lot of novel drugs not scientifically validated till. So it is necessary to validate these compounds from the medicinal plants. It does not have any side effects. So nowadays the academia and industry is moving to medicinal plants for take the compounds from the medicinal plants and use the compound for various kind of diseases. In the last century, many pharmaceutical products were formulated based on the traditional knowledge obtained from various sources. Traditional knowledge, nothing but we can observe the people, the traditional people, what they are eating for some kind of disease, uh, particularly the medicinal plant. This is the traditional knowledge. Traditional knowledge. We observe the uh, our old people always think like this. They did not use any modern system of medicine. So in our grandma and grandfather, all the grandmothers and grandfathers, they used the plant based materials directly. They intake the plant material for cure some kind of diseases. So we this is the traditional knowledge. So we need to observe the traditional knowledge to get the formulation for the pharmaceutical product development. This is the main source. This is the main source we take the points from the traditional knowledge. More than 900 different phytochemicals have been identified as a compounds of food. More than 900 different phytochemical compounds have been identified as a compounds from the food. So many of the phytochemicals are just we need to find again, because many of the drugs not yet find in the food compounds. So it is necessary to find the compounds, phytochemical compounds in our food product. Source of small molecules or drugs. This is the chart showing what are the chemical compounds we are using for naturally. See here. See here, uh, only 5.7 percentage of chemicals we are obtained from the natural product. Direct natural product we are using only 5.7 percentage of drugs. The same time, natural products derived from synthetic modification. These synthetic modification drugs we are getting from 27.6 percentage, and totally synthetic drugs often found by random screening or modification of existing agent. So this is the synthetic, totally synthetic chemicals. We are obtained from the chemical or synthesis from the existing agent chemical. So this is the more percentage we are using for the drug industries. 49.6 percentage we are using for the modern medicine system, totally synthetic drugs. And synthetic drugs, but at the same time it made by the total synthesis, but the pharmacophore was natural product. What is the difference between synthetic and um, pharmacophore natural product synthesis? This one is the pharmacophore is natural, but we are preparing this chemical compound by the synthetic group. This is the difference from both compounds. So only 5.7 percentage of natural products we are derived from the only natural product. So which is necessary to improve these 5.7 natural compounds, because these natural based medicine system does not have any side effect, but these synthetic and semi-synthetic compounds are having many kind of side effects which produce some um, like in cancer disease we are using the chemotherapy chemotherapy treatment, which leads to many kind of side effects like um, hair falling and aging factor. All these side effects happening because of the chemotherapy. But at the same time, the natural products does not produce any side effects, including cancer therapy and other human ailments. It is a universal enigma and nature's pride. It is very difficult to understand the nature system, so it is necessary to understand the nature system to get the benefit from the nature. As I told you, it has boundless powers and bounteous in nature. Many boundless powers are present in bounteous in nature, and it brings priceless wonder cure. Many of the diseases cured by these kind of natural products. So it brings priceless wonder cure and forms barrier to the diseases. So always protecting our human beings from the disease conditions. And these Plant-based products having non-nutrient plant chemicals contains protective disease-preventing compounds. These are the non-nutrient plant chemicals like phenolics, flavonoids, and other terpenoids. These are the non-nutrient plant chemicals which contains protective disease-preventing compounds. And nutraceutical and phytochemicals are similar in function, pointing to prevent all the like we are intake some food material, fruits and leafy vegetables. These have a nutraceutical properties and also phytochemicals. These functions are similar, only pointing to prevention of diseases. So all these natural products having safety, toxically low level and efficiently very potency. So all the natural products are very safety and low toxicity and efficiently potential in regard to disease prevention and management, especially prevention of disease and management. And have a rich source of vitamin E, vitamin B complex, and vitamin C. So this is existing and incorporating of many kind of sources like a vitamin E, vitamin B complex, and vitamin C. So not only these minerals are also presenting in significant quantities in the plant system, like a copper, calcium, zinc, selenium are the important nutrition compounds available in plant system. Selenium is an established antioxidant compound which cure many kind of diseases, which scavenge the free radicals to avoid the disease conditions. And nutraceuticals rich in nourishing, like we are intake many kind of fruits, leafy vegetables, which contains many nutraceutical properties, specific chemical compounds in food, which have vitamins, additives that may aid in preventing the diseases. In world level, the percentage of population undernourished is Russia 3% of people using the natural based products, Brazil 7%, Nigeria 9%, China 12%, Uganda 19%, and India 20%. Here in India, 20% of people mostly depending the plant-based food, particularly in South India. um, South Indian people always go for plant system to get the food, like fruits, vegetables, and other rice material. All these kind of food and medicinally oriented plant products. All all these peoples using plant-based products. So in India, 20% of the people intake the plant-based material. So, number of plant species which have been studied phytochemically for at least one type of biological activity they have. Here, group of the plants like monocots, dicots, gymnosperm, dendrophytes, bryophytes, lichens. These are the many kind. These are the different types of plant system having biological activity, also phytochemical studies. In monocot plants, 1,283 plants have studied biological activity. In the same time, 3,721 species studied for their phytochemical studies. Like the same way, dicot plants, gymnosperm plants, dendrophyte plants, bryophyte, and lichens. All these studied for the biological activity and phytochemical activity. Um, the main division of find the phytochemical agents, phytochemical compounds is bioprospecting. Bioprospecting means exploration of biodiversity. we use the biodiversity and commercially produce the valuable genetic and biological resources. That means collection of biological material from the natural products, natural biodiversity, and analyze the material for their chemical properties, and also evaluate the molecular biochemical parameters, and also evaluate the um, biological activities like inflammation, pain, cardiovascular disease, these kind of activities, which means bioprospecting. What are the significant therapeutic activities having from the plant products like anticoagulant, antifungal effect, and photo-induced modification of nucleic acid, and hypotensive, anti-fever, anti-inflammatory, and anti-swelling, and also many kind of um, cardiovascular disease, diabetic disease, all significant therapeutic activities having in the phytochemical compounds. How the diseases are happening and how it develops from our body system. The free radicals. Free radicals are the main point for creating the disease condition. Free radicals are the free radicals are having unpaired electron in the outer orbit. Here I am showing here oxygen atom. Free radicals are having one unpaired electron. So this unpaired electron trying to capture another electron from our cell system. So whenever these electron system capture one electron from another cell, which continues the chain like reaction. So this chain like reaction creates the uh, reactive oxygens, which create the reactive oxygen species in our cell system, and which produces the disease condition. So generally free radicals are unpaired electrons from oxygen obtaining one more unpaired electron, forming free radical, then affect the cells or lipid around it. So the chain-like reaction affects the neighbor cells, neighbor cells. So it continually going to chain-like reaction and create the disease condition, which caused by pollution system, radiation, and cigarette smoke, sunlight, environmental chemical changes, exposures of metals, biological materials, including food, and chemical reactions that take place in our body, which create the free radical changes. How to control the free radicals? In our body system have some antioxidant system which always working for control the free radical system. These antioxidants are the substances that scavenge the free radical by donating electron from hydrogen atom. Here I am showing the free radical molecule. These free radicals have one unpaired electron in the outer orbital. But at the same time, these antioxidant molecules donate one electron to the free radical system, which scavenge the free radicals. When these antioxidants donate one electron, these free radicals getting normal radical ion. So there is no harmful effect from this radical. Natural um, Basically, these antioxidants are two types. One is natural and enzymatic antioxidants. Balance between the pro-oxidant and the antioxidants in the body system. Also have some synthetic antioxidants commercially available in the pharmaceutical market, like butylated hydroxytoluene and butylated hydroxyamazole. These are the two synthetic antioxidant molecules available in the market. And these are the what are the reactive oxygen species and reactive nitrogen species in our cell system? Superoxide, hydroxyl radical, peroxyl radical, alkyl radical, and hydroperoxyl radical. These are the radicals species present in our cell system. At the same time, non-radicals like hydrogen peroxide, hypochlorous acid, ozone, singlet oxygen, peroxynitrate, and radicals, nitric oxide radicals, and nitrogen dioxide radicals. These are the reactive nitrogen species available in our cell system. Whenever changes happening in this reactive oxygen and reactive nitrogen species, which induces the disease condition. Whenever these reactive oxygen species and reactive nitrogen species does not work properly, at the same time which produces the disease condition in our body system. How these reactive oxygen species and reactive nitrogen species developed from in our body system because of the environmental factor associated as an oxidative damage, environmental pollution, ionizing radiation, and UV radiation, and harmful substances like toxic substances, xenobiotics, and smoke, and taking alcohol. These are the environmental factors which is associated for the oxidative damages and produces the reactive oxygen species, nitrogen species in our cell system. Classification of antioxidants is enzymatic antioxidant and non-enzymatic antioxidant. Enzymatic antioxidant means superoxide dismutase, glutathione peroxidase, catalase, glutathione reductase, and glutathione transferase. Non-enzymatic antioxidant alpha tocopherol. These are the non-enzymatic alpha tocopherol, nothing but vitamin E. Vitamin E is called as alpha tocopherol, and beta carotene, ascorbate, glutathione, selenium. These are non-enzymatic antioxidant compounds. What is the mechanism for the antioxidant action? The main mechanism is inhibition of generation and scavenging capacity against reactive oxygen species and reactive nitrogen species. At the same time, reducing the capacity level and chelation of metal ions and um, activity as an antioxidant enzyme and inhibition of oxidative enzyme. So these are the main mechanisms involved in the antioxidant action. Like this. Here, this picture shows that antioxidants oxidize the antioxidants and reduce the reactive species, reactive oxygen species, and reactive nitrogen species. Also, scavenging. Uh, how we can analyze this the antioxidant activity by some laboratory assays which proves the radical scavenging activity like DPPH assays, scavenging of 2,2-diphenyl 1-pityl hydroxyl radical, one of the best method to analyze the radical scavenging activity, DPPH assay. Another one is ABTS, ABTS cation radical assay, and ferric reducing antioxidant power assay, choline equal to reducing capacity, and total reducing capacity, estimated by electrochemical method. These are the main assays to analyze the free radical scavenging activity, and also reactive reactive adjacent species and reactive nitrogen species analyzing assays. These are the another assays, enzymatic assays we will use in our laboratory assay. Peroxyl radical scavenging capacity, superoxide radical anion scavenging capacity, hydrogen peroxide scavenging capacity, hydroxyl radical, hypochlorous acid, singlet oxygen, nitric oxide radical, and peroxynitrate scavenging capacity. These are the enzymatic assays for um observing the radical scavenging activity in the laboratory way. Also, uh, The antioxidant property of the plants which which improve uh, the extract of the plant material having antioxidants which helps to reduces the other ailments like analgesic, antipyretic, and the anti-inflammation. How it is possible? Whether all the extract of the plant-based products having antioxidant capacity, it definitely have some biological activity like analgesic, anti-inflammatory, antipyretic, and wound healing studies, and also diabetic studies and cancer studies. So, whenever the antioxidant molecules are available in the plant medicine, medicinal plants, which also have the biological activity like these kind of analgesic, anti-inflammatory, wound healing, and bio and anti-diabetic activities. All these studies are biologically uh, evaluated from various kind of assays. Especially in the anti-diabetic studies, we are using alpha amylase enzymatic inhibition assay, alpha glucosidase enzyme inhibition assay, and enzyme and gene expression in diabetic induced cell lines. In vivo studies, we are using streptozotocin in induced diabetic rats and using gene knockout rats. These are the main methods we are using for the study of anti-diabetic activity. Like wound healing, we are used like burn wound, excision wound, incision wound. These are the three type of method we are using for the study of wound healing activity. And anti-inflammatory studies like membrane stabilized method and protein denaturation and carrageenan induced inflammation, exalbumin induced inflammation, cotton pelo. These are the main methods for analyzing anti-inflammatory activity. An analgesic activity like hot plate method, tail flip experiment, acetic acid induced with test, formalin-induced polyethylene paste, and the yeast-induced pyrexia, like an antipyretic activity. These are the main methods we are studied for the analgesic and antipyretic activities. And after the biological activity is finished, we are going for the glucose parameters like blood parameters, biochemical assays, like serum parameters, serum estimation, serum triglyceride level analysis, serum cholesterol level, and glycogen estimation. All these blood parameters we need to analyze for the biochemical. Um, Here I am showing the food products have what are the phytochemical agents. Here alien vegetables like garlic, onion, cheese, and leeks have the phytochemical compounds like allyls sulfides. The same cruciferous vegetables like broccoli, cauliflower, cabbage, and Brussels sprouts and kale, turnip. These kind of vegetables have indole, glucosinate, and sulforaphane compounds and isocyanide and diols. These kind of phytochemicals are available in these cruciferous vegetables. The same kind of Solanaceae vegetables like tomato, peppers have lycopene compound. The same kind citrus fruits have monoterpene, and other fruits like grapes, berries, cherries, apples, and watermelon, pomegranate having allergic acid, phenol, flavonoids like quercetin compounds. So I already told these phytochemical compounds are enormously present in the plant-based products, plant-based vegetables, plant-based fruits. So it is necessary to validate scientifically to improve the pharmaceutical products. Utilization of global vegetables like tomato, brinjal, broccoli, onion, and cabbage and chili. All these global vegetables have a lot of medicinal properties like this bitter gourd, leaf vegetables, bean and spinach, moringa, and cowpea, and okra, and bottle gourd. These are the indigenous vegetables. How to promote the indigenous vegetables? We can grow the indigenous vegetables by women and children through the human and school gardens and improve the capacity building activities such as field days, farmer field schools and demonstration plots, and posters and mass media and national campaigns. So these are the main systems we can promote the indigenous vegetables. And see this project showing the agriculture and industrial based plant-based products, agronomic plants and food quality plants and ornamental plants and plant defense mechanism. So these product-based plant plant development, which helps to develop the and industrial as well as commercialized product. Develop the product like industrial and commercial products, like a pharmaceutical products, and also uh, natural dyes industries and oil industries. So the plants which is developed from the industries and also pharmaceutical products, which helps both uh, both units of the system. And natural products and medicinal plants approaches we are going to see now. Herb- Herbs as the alternative medicine system for the modern medicine system. Modern medicine system has been replaced for around 200 years only. But at the same time, natural medicine system has replaced for over 4,000 years ago. Herbal medicine are more safe, effective, compatible, and well tolerated. More than 25% of modern drugs include chemicals originating in plants. Example: aspirin, cocaine, and carar, and digoxin, and ephedrine, and podophyllatoxin, sena, theophylline, and tobacco, cocaine, and toxal, vincristine, warfarin, many kind of drugs originating from the plants. And global herbal market, 62 billion US dollar world herbal market. In world herbal market, 62 billion US dollar per year using in the natural products. Mainly, these compounds cure the metabolic disorders like cardiovascular disease, diabetes, and obesity. Uh, Metabolic disorders causes disturbance in the normal chemical process in our body system and will result in the different symptoms depending on the particular disorders. The process of converting food into energy is known as metabolism. These metabolic functions also govern the transport, storage, and distribution of energy throughout the body. The myriad of metabolic functions are constantly occurring in the body of any living organism as the cells work together to keep their parent organism healthy. A major part of a healthy metabolism is the generation of enzymes which break food down into energy and handle the transport of the energy. Most metabolic disorders are related to the enzymatic malfunction. Yes, these enzymatic malfunctions develop the more most of the metabolic disorders like cardiovascular disease, diabetes, obesity. These enzymatic functions like glutathione peroxidase, SOD, superoxide dismutase, and alkaline peroxidase. These are the enzymatic activities. When uh, these enzymes changes into in it changes made in these enzymes which which develop the metabolic disorders. Diabetes mellitus is one of the major metabolic disorder. About 285 million diabetic cases worldwide, accounting for 7% of the world population. The World Health Organization, who projected that without urgent action, diabetes-related deaths will increase by more than 50% in the next 10 years. Uh, are you hearing my audio, my Anna Maria?
Yes, we are hearing you.
Okay, okay. Do you understand the slides?
Yes.
Okay,
Pessoal, alguém tem alguma dúvida, alguma coisa? No, I think it's very clear.
okay. Then I will continue.
Okay.
Many of the natural products like chemical compounds we are identified by some instruments like the GCMS, LCMS, HPLC, like this kind of instruments. These are the main instruments which is helpful to identify the chemical compounds from the plants. Techniques commonly used for the isolation and identification of phytochemicals is GCMS, identification of volatile non-polar compounds by comparing the mass spectra in the standard library. LCMS, identification of non-volatile polar compounds by comparing the mass spectra in the standard library. And also HPLC, high performance liquid chromatography. This is also one of the identification technique for non-volatile polar compounds by comparing the retention time with the standard compound. So these are the main uh, techniques commonly used for the isolation and identification of phytochemicals. And uh, whenever the free radicals changes occurring in our body, these damages make many system leads to serious long-term complications like eyes, brain, cerebral circulation, kidney kidney function failure, and cardiovascular diseases. These kind of problems arises from the radicals problem. Some of the plants I'm going to explain for you. What are the chemical compounds present in this plant and how it is used medicinally and also traditionally. Okay, I'm going to explain some of the plants for you to. This is the plant Dioscorea obstifolia. This is the family Dioscorea. Tamil name you doesn't know. It is not necessary. Medicinal uses like leprosy, cancer, antiseptic, ulcer, chronic diarrhea, asthma, and dry cough. And here the edible part is trunk and also vegetable. It is a leafy vegetable and bioactive compounds like triterpenoid, vitamin C, vitamin B, alkaloids, steroids, terpenoids, sulfuric acid. All these compounds are available in this plant, which is used mainly for leprosy, cancer, antiseptic, ulcer, chronic diarrhea, asthma, and dry cough. Another plant, Gravia tetrotricha. This is Tiliaceae family. This is the plant from Tiliaceae family, and it also have many kind of medicinal uses like cough, epilepsy, and urinogenital diseases. And the bioactive compounds present in these plants are phenolics, alkaloids, and steroids. Here, the edible part is fruits. Uh, Leaf is vegetable also. Leaf also used as a vegetable. Another plant is Clapsis hemiltoni. This is Asclepiaceae family member and medicinal uses like skin diseases, diarrhea and nutrition diseases and blood purifier. It is a good blood purifier and also used for curing central nervous system disorders. Many kind of bioactive compounds available in these plants like 2-hydroxy-4-methoxybenzyl dehydrate and methyl salicylate, benzyl alcohol, and this vanillin, ketone. resinol, sterol, saponin, and tannin. These are the main phytochemical compounds which is available in this plant. Here also, leaf is vegetable edible stem. Another plant, Fluctoria indica. This is Fluctoraceae family, and the medicinal use like snake bite, diarrhea, cough, arthritis, bacterial throat infection, and bioactive compounds present in these plants are uh, beta cytosterol and beta cytosterol, glycopyrinosides, ramonosides, and phyto and butyroplactone, lignin disaccharides, and flocotine and coumarin. These are the main compounds available in this plant. Fruit is an edible part of this plant. Leaf is also vegetable. And Carissa carandus. This is another Apocynaceae family group of plant. This also having medicinal uses like anti-diabetic, anti-cancer, antibacterial, and hepatoprotective activity. The main bioactive compound is carizin and beta phytosterol. Here also leaf is vegetable. Fruits also. Fruit is an edible part of this plant. And another plant, Alangium salvifolium. Alangnaceae family. The medicinal uses like anti-hypersensitive, antidote, anti-inflammation, and anti-hemorrhage, antibacterial, anti-diarrheal activity, cytotoxicity effects. All these effects having this plant. And by bioactive compounds like phenolic glycoside, alkaloids, steroids, all these compounds are available in these plants. Here, nut is fruit is the edible part of this plant. And Kakliku, orchioids. This is Hypoxidaceae family, and which is mainly used for Alzheimer's disease and jaundice, venereal diseases, and the bioactive compounds like Carclicoside A, B, C, and B, and carcinogen A and B. Here also trunk is a edible part. These are all these plants are we are collecting in the traditional knowledge level. So this is only the traditional knowledge level of trees. It is necessary for the scientific validation. So it is necessary to go for biological activity in the laboratory manual. Another plant, Cyanotis tuberosa, Comulinaceae family. It also has continuous fever, diabetes, medicinal properties like bioactive compounds like anthocyanin, flavonoids, carotenoids, and vitamin C is available in this plant. So, like the same kind of plants, Caroluma diffusa and Anthium dicocum and Plantoca asiatica. Here, these plants have many medicinal uses like anti-inflammatory, antibacterial activity, antiseptic, anti-tissue, cardiac disease, cardiac activity, and hemostatic activity, repair of joint like rheumatic arthritis problem, and hypertension, reducing the hypertension, rheumatism, seeds and bulk laxative and smooth the irritated membranes. So many kind of medicinal uses having these plants. This is also some of the traditional people um, people are living in a hills area, so they use these plants for um, these kind of diseases. Main bioactive compounds like phenyl propanil glucoside, phenolic linalool. These are the main bioactive molecules. And Poperia dulcis. Poperia dulci, another one of another plant, also having many medicinal uses like dysentery and fever, gonorrhea and headache, jaundice, snake bite, stomach problem, and toothache problem, like hypertension, and antioxidant, antimicrobial, analgesic, and anti-inflammatory activities. and the main bioactive compounds like uh, scoparinol, flavonoids, terpenes are available in these plants. So many kind of plants are available in our biodiversity system. Here one is Prianthema, Decantra, and Hippophobia remanoids. These are the another plants available in the biodiversity with many kind of activity. Here, here also, Elium viburnum and herbal applications ingredients like Origanum vulgare and Curcum curvi and Funiculum vulgare. These are the of appetizer plants we are using before intake of food. Here I am showing Achillea millefolium. This is Astraceae family. The plant extract has strong insecticidal properties and is used against skin infection, and powdered leaves are given against stomachache complaints. Main secondary metabolites is these monoterpene and sesquiterpene compounds. And Dactyloryza. These are Orchidaceae family. Here also roots are made into powder and given twice in a day for 1 month or more against urine and kidney problem. The tubers are considered energetic, health improving and also recommended for weak people. Here the secondary metabolites, main secondary metabolites, tannin and flavonoids, and photophyllum hexandrum. Here also secondary metabolite is phyllotaxin, which is having radioproductive agent. Aconitum heterophyllum. Aconitum heterophyllum is found in the alpine and subalpine Himachal Pradesh in India. The roots of Aconitum species contains bitter diterpene alkaloids. These diterpene alkaloids are used to treating a number of ailments like fever, diarrhea, hysteria, throat infection, and dysphagia, and abdominal pain and diabetes. So many of the plants from the biodiversity have lot of medicinal properties. Ficus amplicium, another plant which have many flavonoid compounds and phenolic compounds. In my laboratory, in Indian laboratory, one of my PhD colleague, he is taking this plant Ficus amplissima for diabetic studies. Biologically, diabetic activity he was done in this plant, and this plant showing lot of diabetic activity. And rhodiola implicata. This is uh, radioactivity compound, uh, rosin and rosamine, salitroside flavonoids. These are the radioactive capacity compounds. These are mostly available in high altitude region like 4,000 to 5,000 meter altitude. And Borassus flabellifer. This is the uh, this is the fruits of the Borassus flabellifer tree, and we can eat this fruit directly. And this is the rhizome of the plant. All these plant pods having medicinal properties like jaundice, anti-inflammatory, and antimicrobial activity. So the kernel has nutritional status, galactose, galactomannan, and polysaccharides, and fruit pod shows the carotenoids and flavelliferins. Seed should shows carbohydrate, fiber, calcium, magnesium, and iron nutrients. Um, we are patent this uh, powder we obtain from this kernel uh, to improve the nutritional properties of the nutritional properties. And Merinia tridentata. This is another plant of Convaviaceae, which is available in our Tamil Nadu and other places. The plant is considered bitter, astringent, tonic, and used in rheumatic arthritis and pile swelling, urinary disorders, wound healing, anti-inflammatory, analgesic, antioxidant, and anti-arthritic activities. The secondary metabolites like flavonoids, diosmitin, luteolin, and pyrrolidine alkaloids and ergosine alkaloids are available in this plant. And gymnostatium fabrifusion is another plant. These also have many kind of secondary metabolites like flavonoids, diosmitin, luteolin, like compounds. These, especially the root system of these plants, local people used fabrifuge and applied to the tongue to remove blisters and sores. Contains bitter principle of the resinoid nature and small quantities of tannin and sugar. This is the passiflora species, like maracuja. This is the banana passiflora, Passiflora mollissima. These Passiflora species are very popular for their fruits, passion fruit. Especially all the kind of fruits are edible in this plant species. Um, Also, the leaves also used as a tea from the leaves. It has been largely used for folk medicine. These Passiflora species also having sedative, diuretic, asthma, hypertension, and skin disease. Also used as an antioxidant agent and nutritional agent. Many of the Passiflora species having glycosides, passiflorane, flavonoids, ascorbic acid, cyanogenic glycosides, apigenin, isobactasin, and orientin and C-glycoside flavonoids, flavum, lysine, flavonoids, alkaloids, hexanol and hexanol and iron, and passiflora. These are the different kind of species in the Passiflora species. In our Brazil, we have a maracuja fruits, right? The maracuja fruit, the name is Passiflora ligularis. I did one of the antioxidant and anti-diabetic activity in Professor Narendra's lab. That paper also accepted in Food Research and International Journal. That Passiflora ligularis, maracuja have a lot of diabetic activities. We observed this activity by alpha amylase inhibition assay and alpha glucosidase inhibition assay and also antioxidant activity assays. All these activities shows enormous amount of effect in these plants. So some of the biological activities uh, we need to do in further future experiments for these maracuja plants. These are the major phytochemicals of the Passiflora maracuja plants: alkaloids, phenol, glycosides, flavonoids, and cyanogenic compounds, and classifloricin, and polypeptides, and alpha-pyrin. These are the main compounds which is available in the Passiflora species. And some another species, Monocoya vaginalis and Pothos candens and buckwheat and Psychotria nilgiriensis, Psychotria nilgiriensis. So I am explaining many of the plants which have many phytochemical compounds which is used to cure many kind of human ailments like properties like cardiovascular, anti-diabetic activity, anti-inflammatory activity, and analgesic activities, and also hepatoprotective activity, radioprotecting activity like this. Also, all these kind of medicinal plants having lot of secondary metabolites like phenolics, flavonoids, terpenes, saponins, alkaloids, like this kind of compounds. This is the main point. All the plant-based materials having lot of phytochemical compounds which definitely helps for the human beings to cure the diseases. In our laboratory also, in our Professor Adriano laboratory and also Lucindo laboratory, we are using many of the terpene, monoterpene, septuterpene and saponin compounds and also plant-based phytochemical compounds. We make the inclusion complexes between the pure compounds and inclusion complex between the cyclodextrin molecule. So the both compounds make the uh, inclusion complexation makes which improves the bioavailability nature of the chemical compounds. So when we are making the inclusion complexation in the chemical compound, this inclusion complexation which develop the bioaccessibility and bioavailability of the when the drug enter into the our blood system, it directly absorb in our blood system easily. Though inclusion complexation which helps uh, intake the chemical easily in our blood system. So all the phytochemicals we use in our laboratory to make the inclusion complexation for developing the pharmaceutical drugs in future and modern medicine system. Especially nowadays in the COVID just now we also it is very difficult situation in the worldwide. So we need to find some new modern drugs for avoid these kind of disease systems. So especially in our lab, we are using inclusion complexation. So we can find some new uh, chemical incorporation for developing the new drugs for various diseases like uh, new invented diseases like COVID and other HIV and many kind of diseases. It is necessary to find the chemical compounds with easily accessible for the blood system. Validation of phytochemicals. Phytomedicinal compounds identified with bioactivity are tested with validation. Validation means nothing but we try to find the phytochemical compounds with biological activity. Scientifically, we need to prove. Scientifically, we need to prove like how we need to work the experiment and publish the article in a reputed journal, which helps the validation of the compound. It is which helps the validation of the compound to show the activity, how the compound active for all the diseases. So 120 distinct chemical substances derived from plants with validated commercial application. Just 120 distinct chemical substances are derived from the plant system now, which is validated commercially and for the different application. It is not a good amount of chemical compound. We have a lot of chemical compounds in the plant system. So we need to identify, also isolate the compounds from the plant products and make the complexation with the cyclodextrin um, and other materials to improve the efficacy of the plant-based products. We need to validate these scientifically and prove the compounds, prove the compounds with biological activity. Uh, Drugs required masses. Why the drugs are required masses? Because of the uh, growing population in our world, it is necessary accessible of drugs and affordable amount of drugs and also assured safety of the drugs is necessary. So it is necessary to required masses of drugs we need to produce for all the accessibility, affordability and assured safety. Do they fulfill the requirements? Definitely, the plant-based products fulfill the requirements of the biological activity and also marketization and develop the products. Um, all these are fulfilled by the plant-based products, especially natural products. I already told you why the scientific validation is necessary for developing the new drug systems. Here I am showing for providing alternative medicine and supplementary therapies. Cultivation of important wild medicinal plants with stable heritable characters of therapeutic importance. When we scientifically proved that plant will move for the tissue culture laboratory, like kind of laboratory, and the people will do cultivation of the plants easily in the vegetation area. So it is necessary to scientific validation, development of package practice, post-harvest handling of medicine plants raw materials. So when we go for scientific validation, it is helpful to cultivate the plant and develop the plant-based product, raw material, and also which help us make the safety of the plant product and also customer satisfaction, confidence. When we did the scientific validation, people always believe, okay, this product is good product for this kind of disease, already proved by scientific literature, scientific articles. So it is necessary the scientific validation to prove the chemical compounds have biological activity or not. Chemical markers for medical plants. Uh, when we prove these scientifically validated the biological activated activity compounds is uh, always come for commercial markets. So when we go for the scientific validation, then final products come for the final marketization, especially the chemical markets for medicinal plants. Biological activity is rare molecular property. The chemical diversity of compounds from medicinal plants is much more than chemical library developed by the scientists. So the bioactive compound specific to that species is called chemical markers. The bioactive compounds, here we can see the bioactive compounds specific to that species is called chemical markers. Like chromatographic techniques such as high performance thin layer chromatography, high performance liquid chromatography is used in chemical profiling of medicinal plants. I already told these. Uh, these are many of the techniques which is used to identify and also isolation of these kind of compounds, which is helps to uh, analyze the purity of the compounds, purity of the compounds, and Nat- novel bioactive substances, naturally available products discovered from medicinal plants, have provided numerous clinical use medicines. These natural products are important source of the biologically active compounds and have potential for development of novel drugs products. It is important to ensure their conservation and sustainable utilization. We need to conservate the medicinal plants for sustainable utilization. Sustainable utilization means uh, for the growing population, we need large number of plant products. So that means utilization, sustainable utilization. So definitely we need to conserve the medicinal plants in a vegetation area for sustainable utilization. The evaluation and authentication of medicinal plants for better healthcare system, and many reputed medicinal plants are critically endangered in many regions and also having tremendous potential for bioprotection and constraint. Lack of good field botanists, lack of cooperation between taxonomists and biotechnologists, lack of interest in the field of studies and funds. So definitely the more care we need to put in development of the plant-based pharmaceutical industries, plant-based modern medicine system, therapeutic medicine system. We need to take more care for constructing the new system of medicine system, especially 21st century, wasteful with excitement and novelties in improvement of human health based on herbs. Many of them yet to unexplored and untested. Yes, many of the plants still now untested unexplored. In Brazil also have a biodiversity, very big biodiversity like Amazonia. This Amazonia is one of the biggest biodiversity in the world, which produced more amount of oxygen for the world. More than three-fourths of the oxygen level produced from the Amazonia forest. So this is the main point. So the conservation of forest, the conservation of medicinal plants, and conservation of natural products. It is necessary to take more care for find mm, many of the better and good products we develop from the biodiversity in our nature. So it is necessary to uh, study the plants and chemical compounds and necessary to explore these kind of medicinal plants and bioactive compounds for many kind of bioactive compounds with human ailments. So it is necessary to explore all these kind of biochemical compounds and medicinal plants in future also. It is helpful to reduce our risk factors. Okay. Phytochemicals are known to have wide range of economic importance and applications to human health and welfare. So it is necessary to know it and use it and save it. So conservation of forest, conservation of biodiversity, it is necessary to know how we can conserve the forest, how we can protect our biodiversity, and how we can use the biodiversity for our regular life, and how we can save it. It is necessary to understand the biodiversity system, and we can go for further healthier life. Okay, thank you, Ananda. Okay,
Thank you, Sarah.
yeah, thank you
Pessoal,
very much, Ananda.
thank you. It was amazing. Pessoal, vocês têm alguma dúvida, alguma coisa para perguntar para o Sarah em relação à apresentação dele? People are saying congratulations and thank you for the presentation.
I don't know how
Congratulations, amigo.
Obrigado, amigo, obrigado. Eu não sei como eu fiz these slides, the full screen mode. I don't know. I doesn't know. I forget this option, so I'm not able to get the full screen mode. So I'm very sorry for the inconvenience. Okay.
Okay, but besides that, we saw all the slides and the presentation. We
Okay.
could solve. Okay, don't worry
Okay.
about this.
Okay, I will send you the Maria. I will send you the slides for review, and you
Okay,
can you can send to all our friends for further clarifications. Okay.
okay.
If you
Thank you, thank
if you have any questions, please ask me. Tenho perguntas, amigos.
you. No,
Algum
Posso fazer uma
perguntas?
pergunta em português, Ana Maria?
Pode, Gustavo.
Saram, você
Pode.
poderia falar só um pouquinho, se você vê muita diferença entre a pesquisa aqui no Brasil e na Índia, como você vivenciou os dois? Acredito
Hum,
que eu fiquei um pouco curioso. Você poderia falar só um pouquinho?
pode falar.
did you understand, Sarah?
Para você falar, Saram, o que você viu diferente aqui com relação à pesquisa na UFS e com o que você fez na Índia, com relação à estrutura,
Totalmente
a
diferente,
execução da
amigo.
pesquisa?
Sim. Aqui, totalmente diferente, amigo. Na Índia, pessoas This was so, um, how to say, um, select the plant material.
Seleciona o material
Material selection of the
de
plant.
pesquisa, né? Seleciona o material de
So
pesquisa.
aqui tem pouco actividade, like biological activity, like antioxidant activity, and some of the animal studies we have here. But in our Brazil, we are using modern medicines, modern techniques like identification and isolation technique we are using in our laboratory. It is very modern. So here in India, it is available in Indian University also. Tenho na Universidade da Índia também, da técnicos. Mas uh, find these techniques is very difficult in Indian University. Okay, the laboratory, the colleagues um, mm, not have this kind of techniques. So it is it is different from our Brazil and India. It is the one of only one different. We have lot of instrument in our Brazil, but in India the instrument system is very less. But the same time the some of the how to say particular instituto particular, né? Instituição de particular tem todas instrumentação para analisar de atividade. Entendi, amigo?
Muito obrigado, amigo. Entendi sim.
Mais alguém, pessoal? Congratulations, Saran, for the presentation.
Thank you, Ana Maria.
We like it very much. I will stop to record now, okay?
Okay. Okay, Ana Maria.